Good morning, everybody, and welcome to the CSCR. Today we have a lecture by a most esteemed speaker in the domain of basic science research in the area of ophthalmological sciences. I'd like to proudly introduce to you Dr. Indumathi Mariappan, who has completed her doctoral and her postdoctoral research training at the CCMB in Hyderabad in the year 2008. Subsequently, she's done her further training in the areas of examining the role of nuclear matrix proteins such as the A-type lamins and regulating muscle differentiation and signaling mechanisms and regulating muscle stem cell quiescence and reactivation. Subsequently, in 2008, she joined the stem cell biology lab at the LV Prasad Eye Institute in Hyderabad as a full-time research faculty and continued there until 2025 before she joined the INSTEM Institute in Bangalore. Dr. Indumathi's lab at BRIC INSTEM is involved in basic and translational research towards addressing the problems of retinal and corneal diseases using different sources of adult and pluripotent stem cells. Her group is currently exploring the applications of human-induced pluripotent stem cells to generate 3D corneal and retinal organoids and genome editing technologies to create gene knockout and mutation corrected patient specific stem cell models to understand ocular disease, pathobiology, and to develop cell and gene based therapies for certain inherited ocular diseases. I think that's a fantastic repository and a background to Dr. Indumathi. This is really broad-based, fantastic research in the area of ocular cell biology and genetics. We look forward to your talks. Over to you, Dr. Indumathi.
Very good morning, everyone. And first of all, let me thank Dr. Nihal and Mohan. I didn't see Mohan. Oh, okay. Mohan for giving me an opportunity to come and share some of our experiences in the ocular space amongst you today. And so for many of you, I'm a very familiar face here, but as Nihal said, very recently I moved from LV Prasad to INSTEM. And definitely looking forward for an extended collaboration here with the ophthalmology team. So that's my whole purpose of being here, to share and introduce you over what we have done over the years. Today I'll be talking about understanding some of what we have done in the space of inherited eye diseases and towards developing newer therapies. So I don't have to introduce this for the group here. So our research is mostly focused on the front part of the eye, which is cornea, at the same time on the backside, which is really a lot of things are black box here. And so broadly, the area of the lab is to use some of the stem cell based model system, particularly patient specific stem cells, to understand the disease biology, because we know several genes, over 300, have been implicated in many inherited diseases. We really don't understand how each of the genes function and why these gene mutations could affect specifically eye and that to particular organs or tissues within the eye. So that's the broad basic science interest in the lab. So we are developing many disease models. Currently, my lab is working on several of the genes. I'll give you one example to give you a glimpse of what we are doing. And many stem cell models, we have also been developing some zebrafish models in the lab earlier. And using this comprehensive in vitro and in vivo model, we try and understand this disease biology. And parallelly, from the translation point of view, we have been working towards developing cell-based therapies, not gene-based therapies, how we can generate healthy iPSCs, and from there we can generate retinal cells for allogenic therapy. And can we combine gene editing if you know what the mutations are involved? Can we correct these mutations in the patient-specific stem cells, generate cells which can be tailored for the patients? That's another area. So I'll give you examples of what all we have done so far. So in the space of disease modeling, I'll just give you one example as for the retinal dystrophy, retinoblastoma model. So the mutation in the gene RB1 is the main cause of tumor formation, but it's really not unknown why a gene which is important for cell cycle control for any nucleated cell should show a phenotype only in the eye. Why retina is more prone for getting transformed because of these mutations, something that we were interested. So we have generated many patient-specific stem cell models and gene knockout models. And now we know what is happening is the current theory is the photoreceptors, particularly the cone precursors of the cell of origin, and these are the cells that will de-differentiate, proliferate, and give rise to tumor. But what we find with the stem cell models that doesn't have express in RB, we now know that there are two ways of differentiation that happens when the progenitor differentiate to form the retina. So there are six different neurons. The first two types, the RGCs, horizontal amacrine cells, emerge first in the first way of differentiation. There's an intermediate precursor which then go on to give rise to a late stage precursor which will differentiate into the late cell types. What we think is that it's not the late stage progenitors that is differentiating you in tumors. You have an intermediate precursor which is predominantly formed in the absence of RB because RB is not dependent, the cells is not dependent on RB here. So whether the RB is there or not, cells make these intermediates. But if the RB is not there, then this late stage differentiation doesn't happen. The cells get stuck up here. And these are the progenitors that really expand. So there's a precocious differentiation in the absence of RB. So most of the stem cells gets lost. They become progenitors here, and this accumulates, and these cells are now prone to a lot of transformation that give rise to tumor. That's our understanding now. So how did we came to this understanding? So we have generated these models. We have normal iPSC-derived retinal organoids. We make retinal stem cells and then make organoids out of them. And then you have patient-specific one. Patients always are heterozygous. We have the heterozygous patient-specific lines. And use CRISPRs to edit and knock out both the alleles of RP, and you have double knockout line. So you generate all these tissues and follow them over differentiation. These retinal organoids mature in the dish over a period of 90 to 100 days. So you follow them, and what we observed. So to just give us a crisp idea, so you can just follow the red staining here, which marks the stem cells. You can see that in the double knockout, the red stem cells are precociously lost, even within 55 days. So these cells are lost. But the intermediate precursors, which are labeled in green, are really proliferating. They are expanding abnormally, and they don't laminate properly. They just lost their positional information in this case. So these are the cells that are now prone for undergoing transformation. So that's broadly an idea about what stem cell models can do, which we can't really follow at the fetal stage in an animal or in a human system. Now, moving on to the translational aspect of it. So we started with the corneal program to begin with, in the sense when we differentiated, so you can generate corneal organoids. So cornea, you know that it is not, again, as a simple structure. You have a stat-based squamous epithelium, you have a stroma, thick stroma, and an endothelium. Each of these cell types originate from different lineages, whereas ectoderm contributes to the surface, and your neural crest cells, the different lineage of cells contributes to the stroma and the endothelium. So when you make stems, you derive these corneal structures from stem cells. All these structures come together simultaneously, and they self-organize to form a complex structure, which is now possible. So, but being at LV Prasad, when I joined the institute, I had the opportunity to work with the clinicians who are developing adult stem cell therapy for the corneal surface reconstruction. We know that for the epithelium, there are stem cells that are sitting at the periphery in the limbal region, and these tissues were harvested to generate grafts, epithelial grafts in the lab. So there are conditions like you have LSD, which is the surface defects. You have endothelial defects, endothelial dystrophy, which is corneal, where also the cornea becomes opaque. And you have the stromal defects, which is also inherited. There are many genes involved in the stromal dystrophy. And all three layers have either surface defects which can be either due to burn or injury, whereas these are inherited diseases. So you have variety of spectrum of diseases that affects the cornea as well. And it's for the epithelium, the therapy is already available. So where you harvest cells from the periphery and you can remove the panus tissue on the surface and you can culture them in the lab to generate a limbal graft, which was what was pioneered by Dr. Sangwan and Geeta Vaimuganti at LV Prasad. And my lab has established this protocol for generating complex retinal tissue grafts, corneal epithelial grafts on amniotic membrane as a metric. Basically, you have amniotic membrane, which has its epithelium, which is again a vascular tissue. Remove the epithelium and then grow these limbal graft tissues biopsies on the surface to establish a monolayer graft. So this tissue goes to the clinic for transplantation. So here is a panus tissue. They have to remove this is post-transplantation because it's only an epithelial defect. So the moment you transplant the epithelial cells, you regain the transparency of the cornea, and the success rate was reported to be 76%, over 70%, you can say. So what happens in case of deep corneal defects? So patients go for keratoplasty. And also for corneal endothelial deficiencies, where you have it's because of endothelial defects, inherited problem, then again you are totally dependent on full thickness corneal graft or endothelial keratoplasty. And also for bilateral cases, if there are injuries, so you take biopsy from one of the affected eye and culture the grafts. And in case of bilateral defects where both eyes are affected, you really don't have the limbal source. So that's where we were thinking that maybe we have to look at alternative sources, not just donor corneas, but also alternative sources. That's where I initiated a work on PSC using PSC stem cells. So can we generate these cells either from ESCs or from pluripotent stem cells? So these unused pluripotent cells are something that you can generate in the lab. You really don't need an embryo. You can reprogram a somatic cells, whether it is a fibroblast or a skin cell or any other blood cells. It can be reprogrammed by putting in certain stemness factors in the lab, and you can generate pluripotent cells, which are very similar to embryonic stem cells. So these cells, once they are generated and well characterized, they can be a source of stem cells for differentiating whatever cell of interest, be it cornea or retina. So we have established methods of differentiating this to eye lineages. So basically, we follow the normal developmental pathway where embryo develops. It gets committed to different three lineages tissues: ectoderm, mesoderm, endoderm. So if an eye and the brain are coming from ectoderm, we push them to the ectoderm lineage and then generate different eye tissues. So basically, it's a stepwise process. You push them towards one step up than the other by incubating the cells with different growth factors. And the process have to be really robust enough that you generate enough and sufficient cell of your interest and do good characterization of these so that eventually you have the cell of interest for whatever downstream applications you want. So this is roughly an idea about how we go through. So you have the stem cells that are like this. You differentiate them like within a month, you get an eye field like structure, just like how the embryo initially forms. You have eye field structures that are first committed, and you get these structures. And from here, you can further differentiate them over a period of about 2 months to 3 months. You get complex eye-like structures. You have this is a eye look as if you're looking from the top to bottom. So you have the cornea, transparent cornea in the center. You have the retinal structures behind the back. And if you isolate them and put them in suspension, you can generate this kind of 3D organoids. So you have nice corneal primordia here. This is a lens primordia. You have the retinal primordia, including, as you can see, this is a staining for PAC6, which marks the entire eye field, which is the master regulator for eye development. So the entire eye globe glows green, and you can also see, should appreciate that even the optic stalk-like structures do get formed in the dish. So this is now possible, and all this is achievable within a month in culture of differentiating IPSCs. So now you have all corneal structures organized, retinal organized. You can isolate each of them from these complex structures and go ahead from harvesting whatever cell of interest, if it is cornea, epithelium, or the stromal cells, or the endothelium from these structures. Similarly for the retina. So we have allowed these structures to mature for the cornea to generate corneal organoids. These organoids are about 2 to 3 mm, not real. These are miniature structures. These are complex tissues having all cell types of interest, but then they are miniature in size. The 2 to 3 mm diameter, you can appreciate even the limbal-like crypts being formed. So it is across limbus to limbus, the size is about 2 to 3 mm. So now you can't use these tissues directly as a tissue source for transplantation, but they can be very good systems for isolating the cell of interest, which are of fetal age. So we use this iPSC-derived corneal organoids to establish a similar graph for the limbal epithelium. So here, this is the comparison of how you get the limbal tissue from an adult, and you take the same corneal organoids as a source to generate a complex graft on amniotic membrane. You can establish similar epithelial grafts for the surface reconstruction of the epithelium. This work we published long back at Telifasa. So now we have opportunities to extend it for the corneal stroma, keratocytes, and also for the endothelium. Endothelium is a bigger challenge because we don't have any stem cells in the adult tissue. So all the development stops at the fetal stage. If there is any degeneration because of inherited conditions, corneal endothelial dystrophies, these cells will die. The only way to replace it is using the donor corneas. Now you have opportunity to harvest sample enough amount of corneal endothelial cells from these kind of corneal tissues and can generate corneal casts. So that's something that I will be looking on further, focusing on corneal endothelial dystrophies, see if patient-specific cells can be derived or allogenic grafts can be established for the corneal endothelial problems. So now moving on to the retina. So retina, again, it's a complex tissue, different cell types, 6 different neurons are there, and you have one epithelium and glial cell, angular glia. So all these cells are arranged in a complex manner, but the most of the cell type of focus is the photoreceptors, which are in the outer layer, and the RP cells, which support the photoreceptors. In patients who are undergoing dystrophy, retinal dystrophy, be it age-related or inherited, there are problems in the genes that are important for the cell functions, and these cells are the ones that are undergoing degeneration. So in most of the therapies, we are focusing on replacing these 2 layers. So this is how the histology of the retina, the cell of interest are P cells, these are monolayer cells, and you have photoreceptors here. I'll skip here. So the conditions I really don't have to explain for this audience. So, but age-related conditions, it's a macular dystrophy. Macula is the region where the cones and almost of the cells, photoreceptors that are important for bright light vision and the color vision sits. And degeneration starts from the center towards the periphery. Therefore, the patient will have a central vision loss and the peripheral vision is remain preserved. Whereas in case of retinal dystrophy, retinitis pigmentosa, the rods are the ones that are there in the periphery gets degenerated, and therefore the patient will have a tendinal vision. So this is a broad spectrum, but either of these conditions can progress to total blindness eventually. And the progression depends on whether what kind of gene that is involved. If it's a congenital condition, children are born blind or they lose vision within 5 years of life. In case of retinitis pigmentosa, there is a slow degeneration where you have a bigger window. So where anywhere between 10 years to 40 years, people lose vision. In case of age-related condition, more than 60, as the patient's olds, they have this problem and they lose the vision. So if you look at the genes that are involved, there are over 300 different genes being reported. Again, many of them are systemic diseases. We really don't know why the systemic genes like splicing factors or the transcription factors which are important for a general function or really creating a retina-specific phenotype. That's something that for many of the genes we really don't understand. So that's where the basic interest lies, to understand the pathobiology of the disease. So, but in terms of therapeutics, there are a lot of gene therapy products that are being currently explored. So one of the products many of you would be aware of already, the Luxturna. For the LCA2, this is for the gene RP65, which is packed into an AAV vector, and now this is FDA approved and it's commercially available. And this is the only product right now available for clinical use. And there are many in the trials as of now. And we also have many trials going on for embryonic stem cells and iPSC-derived cells for cell-based therapeutics. So as I've shown you in the early slide, so we generate the eye fields, and from the eye fields, we generate neuroretinal organoids. So these organoids are through and through structures. You have from photoreceptors till RDCs together in this particular tissue. And you can also generate RP cells. These RP cells, fully mature form functional structures like this, highly pigmented cells can be established within 3 months in the dish. And you can see these organoids during stages of maturation. I think this is at 1 month, and you have cell organoids at 4 months. You can see that over time, these are most precursors. So the organoids is full with precursors at this stage of 1 month, whereas at 4 months, you can see them, they are laminated. You can see photoreceptors developing here at the periphery as it will normally develop in an adult tissue. So these are rods and these are cone stained for our cones here. So you can establish such organoids, and if you're interested in transplanting RP cells or photoreceptors, you can harvest these cells from these tissues and develop a therapeutics. So we have went ahead. We have established a robust method of making RP cells and established it for GMP compliance, and we have approached the regulators for the clinical grade manufacturing. So we have received approval in 2022. So these cells, you can see that what is the dose? Dose is something that everybody is worried about. Here you can see that in one dish you can harvest about 8 million cells, 6 to 8 million cells. And these cells, and if you look at the dose that is required for the patients, I think I put up here. So RP dose is about 10 to 20 million can be produced in a dish, but anywhere between 0.5 million is the maximum dose required for a patient. So you can see that in a small batch preparation, hundreds of patients can be treated with this kind of preparations. So these cells are normally most of the clinical trials that are going on are being transplanted in the subretinal space. So you can take this in small volumes, like say 50 to 100 or 150 microliter. That's the maximum volume that the subretinal space can hold, and they are injected in the subretinal space at the periphery of where the retinal degeneration is going on. So that's how this is done. So the dose, as I said, it's anywhere between, in the trials, people have tested anywhere between 0.1 to 0.3 millions, maximum 0.5 million. That's the dose required per injection. So in India, while we were developing this and we were about to start a GMP manufacturing, our institute has tied up with a company, which is also a nice company, which is making similar allogenic product for RP by iStem Private Limited, which is again based in Bangalore. And they have got the regulatory approval for starting a phase 1 trial in India. And as of now, about 9 patients have been already treated using this product, allogenic iPSC derived RP cells, 3 different centers. And the company has recently reported the outcomes of this therapy, and they showed that there's no serious adverse events in these patients. Basically, the cells are really safe on these patients as of now, and shown in some improvement, about 15.8 letters improvement. And now they got approval to go to the phase 2 trial, and soon they'll be starting phase 2 on a larger group of patients, say about 40 to 50 patients soon in this year. So that's where we are. So the trial has now started in India. Now we have at least 1 trial starting, and there should be more trials coming up, including, and that's where we are also thinking about taking it forward for our program. So while we are developing this, so in this case, the treatment is given, sorry. So it's given for age-related patients who are older than 60. And in one of the patients who are treated with LVP, so they are already immune-compromised patients, they're older, and they have to be put on immune suppression because it's an allogenic cell product. So therefore, the other immune suppression-related complications also do come up. So there, if you are thinking about treating an young children for retinitis pigmentosa or an LCA cases, then the patients have to be put on long-term immune suppression, which is going to be a risk. So that's why we thought, can we also think about developing an alternate way, either to develop an universal donor cells which can have minimal immune suppression requirements, or can we tailor the patient's own cells for a therapy, autologous therapy? So that's where we were thinking about combining cell therapy with gene therapy by combining gene editing in the patient-specific stem cells. So this gene editing can be done either ex vivo, as I said, you make the patient cells and you edit the cells in the dish and then make the cell of interest, which is RP, and which can be transplanted into the patient, which is an autologous, tailored for the patient. Or the editing constructs can be packed into viral vectors or non-viral vectors and can be directly injected. So this is in vivo editing. These are the two approaches. Many trials are going on. People are trying to explore this. So we took up this approach because we are based thinking about cell-based therapy. We had several lines, as I said, we have been working towards developing cell-based therapy. We have developed an array of lines for different conditions. And we took some of these lines. We thought we will initiate some autologous cell therapy. So while we are thinking about gene editing, you really need a gene editor which is really safe. Like in the sense, I want to change one base to other, and I really don't want to affect any other genes in the cell of the patients. So that's where the high fidelity and safe editing system come into place. Though we were developing many gene edited lines in the lab for basic research, we're not thinking about using it for therapy until we came across this paper from this group at IJB, where they have shown that these editors, which are F1-Cas9, unlike the traditional and widely used SP-Cas9-based editors, this particular Cas9 comes from a different organism, but it shows very high fidelity in the sense that it can specifically target regions of interest, and if there are any mismatches, it doesn't recognize. So therefore, it gives a safety issue. So therefore, we thought we would explore this particular system for our editing. So, initial editors where it goes and binds to the gene of a region of interest, it makes a cut, and then it uses the endogenous machinery to repair the region for incorporating the right bases there. But now you have improved editors, which are base editors, which will go and do the edit without cutting the genome. So it only goes and recognizes and sits in the region. The enzyme that is engineered will convert a base to another base. So how does it work? In case of adenine base editors, there is an adenine deaminase. This is the base, adenine base. There is an amino group which will be removed by the adenosine deaminase, which will convert into an inosine. So therefore, when the polymerase comes and reads it, this will be read as guanosine. So without cutting the genome, by making sure that the enzyme will convert the targeted adenine base, it can be converted into a guanine. So such A to G conversion can be done using these base editors. And similar base editors are being explored here more in CSCR also, which are showing good efficiency. So we can explore that further. So we have chosen two of these candidate genes. We had patients who are LCA2 patients with RP65 mutations, where there is a stop codon in this patient with this TAG. We are now trying to convert this A to G using this editing system. Similarly, in another case, which is an LCA12 candidate, where the RD3 gene, there is a splice defect. The splice site is now mutated, therefore normal splicing is getting affected in these patients. Can we now convert back and restore the normal splicing so that the protein will start expressing normally and the cells will be functional? So this is the first example, as I said, this is in exon 9. So we try to convert this stop into the normal tryptophan codon in these patients. So this is a gene that's important for function in RPE cells, because this is a gene that is important for an enzyme that will convert the visual cycle intermediate pigment, which circulates from the photoreceptors to RPE and gets reconstituted and comes back to photoreceptors. So if this gene is defective, then this pathway cycle gets affected, visual cycle pathway gets affected. So therefore, it is important to be functional in the RPE cells. So the idea here is, if you can edit the iPSCs, this mutation, and convert them into RPE cells, the RPE cells should be starting to function. So we did this edit conversion. So in the iPSCs, yeah, we found, let's skip all these details about the editing, but then we have created iPSC lines which are perfectly edited for the mutation. About 100% of the cells were edited. And once you have this line, so this is in iPSC, so you really have to check the proof of concept by making the RP cells. So we differentiate and make the RP cells. You can see that this is the patient line. So all other genes important for RPE is fine. They are able to differentiate, make RPEs, but then this protein of interest is not expressing in the patient line. So once we edited, the base edited lines have shown complete perfect restoration of expression as compared to the wild type normal healthy IPC derived lines. So now this proof of concept is now available, and we have published this work as well. And coming to the second gene, which is RP65, RD3 gene, again, this gene is important for photoreceptor function. So it is a splice junction mutation, and this protein is important for transporting one-leg cyclases from the cytoplasm of the photoreceptor to the outer segment where it has to do the function and establish the cyclic GMP signaling pathways events there in the outer segments. So if this gene is mutated, it will restrict in the cytoplasm, and then the transport function of the retinal guanylate cyclases will be affected, and that gives the phenotype. So it is a splice site mutation. So what we decided, if we convert this, the splicing should be restored. So basically, the exon 2 and exon 3 junctions is what splicing will be affected in this case. So we have generated this being a photoreceptor gene. We edited the iPSCs and then converted them into retinal organoids in this case. Earlier it was RPE cells, here it is retinal organoids. So once we made the retinal organoids, we did this simple assay of checking whether the splicing is restored or not by designing a PCR-based assay, where you can design primers for the exon 2 to 3 junction. If normal splicing happens, then you'll have a smaller transcript that is being formed. If the splicing is still aberrant, then the intron will be included and we have a bigger transcript being a product being formed. So this is what we assayed for. You can see that in the patients. So this is a smaller product, which is a normal size product, absolutely nothing. So the abnormal transcript is being expressed. Whereas in this retinal organoid line, this normal splicing is partially restored. Here in this case, it is a heterozygous edit because we didn't generate an homozygous edit. So heterozygous in the sense that you have both one copy of wild type, one copy of mutant being present there. So being a recessive disease, even if you can achieve correction in one of the allele, you should be able to, in theory, be able to restore function in the cell because you have at least one normal function copy that is being present. So we have now this proof of concept. We have published that work also. Now, as of now, globally, we have for the eye, we have one product which is currently in trial, which is a CRISPR-based product, which is for LCA10. Mohan is already working on this gene here for the gene called CEP290. The mutation here in this is between the exon 26 to 27. There is a mutation in the intron, which causes a splice altering function. It creates a cryptic splice site. There's a cryptic exon being generated here in this region, which totally disrupts the frame of this gene. So here they're using CRISPRs to pack into the AAV, which is AAV5, and then this CRISPRs, double CRISPRs, will be injected into the patient eye directly. And this will create a nick in this region, and therefore it will restore the normal splicing of exon 26 to 27. So this is basically using the cutting of the CRISPRs. I think Mohan is developing a base editing based CRISPR where instead of cutting, you can correct this mutation here so that the splicing, normal splicing will be affected in these patients. So that is where the research is at this stage. So for the way forward, so there's always a confusion, like there's a gene therapy that's people are exploring, cell therapy that's being explored, which is good for the patient, which is feasible for different kinds of patients. So for patients with well-preserved retinal layers, if you're intervening at an early stage where the cells are still there, but we know there is a mutation, and we have developed a therapeutic molecule in the sense whether it is a gene therapy product. So then you can use this approach for patients for gene supplementation in case of gene therapy and also for gene editing. So gene supplementation therapy will work for only recessive conditions because dominant conditions, it will not work. For certain dominant conditions, such as where haploinsufficiency is a problem, where there are two alleles, only one allele is mutated, one is normal. So if there is insufficient protein being produced, and that is the cause for a dominant disease, then in that case, again, gene therapy will work. But in most conditions, it's gene therapy is targeted for recessive therapy. And gene editing, again, gene editing, whether it is dominant negative or it is recessive conditions, it can work, but provided cells of the interest should be aligned. So that's where the patient selection is important, because we really have to target these patients early on. So diseases which have a broader life or broader course of disease progression, where you have, let's say, in case of retinitis pigmentosa, where the degeneration is low over a period of say 10 to 40 years, then these are candidates which are good for targeting for gene therapy. And for patients with advanced diseases where the cells are already grown, then you really can't do much about it. Then cell-based therapy comes into play. And particularly for RB cell treatment, it's now been it's been well established. A lot of proof of concept has been published that you can really do RB cell therapy for patients who are having intact photoreceptors, but only 4 RB cells are gone. So then you can transplant these cells in these patients. But for photoreceptor-based therapy, we still it is a black box whether these cells, even if you transplant them, will they integrate properly? And there's a lot of basic research that need to be done. So again, for cell-based therapy, whether it's autologous or allogenic, we really have to be very careful about choosing the patients for the trial, because here we are focusing on only RP transplantation. We want to make sure that the patient do carry intact photoreceptors for any treatment outcomes to be documented. So the important considerations are, again, mutation screening is important, whether, as I said, for whether it is for gene therapy or for cell therapy, we really need to document these patients very carefully. So we need to start this process, and understanding the disease progression and the optimal therapeutic time window, as I just said, it is very important. And recruiting the right patients for the clinical trial, because we have to be really very stringent about choosing the patients. So provided the trials are ready, if the treatment products are ready, be it cell therapy or gene therapy, when the trials are about to begin, we should know who are the patients that we'll be recruiting. So we have to have lineup of these patients for these trials, for these prospective trials. And suitable immune suppression regimes, if you're thinking about allogenic therapy, and affordable cost, we'll talk about a little while. So for the future research focus, my lab will be interested to develop, initiate clinical trials for some of the allogenic and autologous iPSC-derived RP product, which we have some pipelines established already, and we will be wanting to work with clinicians to establish this process, to work with the regulators to get the process validated and approved for the clinical trials. And we also want to generate and validate some universal donor IPCs, as I said, for some of the allergenic applications. And also, when you're targeting young children, it's good to have immune. It's important to bypass some immune suppression requirements. So we are also working towards developing universal donors for using them as stem cell source for deriving cells for transplantation purposes. And we are also establishing projects on corneal endothelium and keratocytes for treatment of some of the inherited diseases of the cornea. So these are the 3 broad programs my lab will be working on, and would like to interact with the clinicians here who are interested in participating in such projects. So coming to the cost economics, so as I said, one gene therapy product is already available. Luxturna, it's way exorbitantly costlier, $850,000 for one injections. But such an expensive treatment, none of our patients will be able to afford. So therefore, it's important. So most of, if you look at the cost of this, half the cost goes into the IP of the product, and the GMP manufacturing cost adds up to this huge amount of cost that is involved in the production of these products. By having indigenous IPs, by developing these technologies in-house, we will be able to definitely reduce this kind of cost. And now at this stage, based on the few trials that have seen the light of clinical trial, we now think that it's feasible to bring down this cost to about 40 to 50 lakhs per patient. But the challenge is this is also is very exorbitantly expensive for many of our patients. But as part of the rare disease program, government is giving funds up to 50 lakhs for such rare disease treatment. So still we'll be able to meet within that deadline, but that will not cater to the requirement of all patients. So therefore, we have to think about how do we bring down this cost somewhere in this range. So that's the range that many of us would want to see our products coming in. Let's see how we can make that. So I think with that, I will stop. And so, will these therapies work? That's another doubt. Many of the clinicians will have this. So, we are developing such expensive therapies. Will it really help the patients? For as of now, we know that these some of the gene therapies are working, and cell therapy is safe in patients. But whether it will help them in restoring visions or at least slow down the disease progression, we don't know as of now. Only as and when more clinical trials have been done, and we will get to know that. So for which we have to get into the process, have more trials, and only then we will learn from there. So therefore, this unknown aspect should not stop us from going progressing and getting us ready for the futuristic therapies. So we have to get started. We need more trials in the country too, and prevention is better than cure. So while we're talking about developing cell and gene-based very happening technologies, it's also important to reduce the burden. So we can't, because our India is a huge population, we can't cater to the needs of millions of patients. So therefore, our the testing and counseling efforts have to be really strengthened so that we can reduce these people in the population, so that the number of patients that require the treatment can be reduced. So this requires stringent uh, pre-implantation genetic testing, prenatal testings for phenotype genotype correlations. At least even for the trials to be ready when the clinical trials are initiated, we should know who are these patients, because we need this database. Without this database, we're not going anywhere. So therefore, it's important for the clinicians also to get interested in gene testing, at least counsel the patients to get into, if required, if you see a pedigree that is there is strong inheritance being seen. It's important to counsel them for genetic screening, and there are companies now providing testing services. Say, for example, MedGenome is now having a complete, comprehensive ophthalmology panel being ready. It's been available anywhere in between 10 to 20,000. I don't know now what is the exact cost, but it should be negotiable. We can talk to them and can bring down the cost. So if patients are interested, we should be counseling them to go for genetic testing so that we know what is the gene involved at least. So when a gene therapy comes, so if Mohan or any of us are developing gene therapy, then we should know who are these patients. So any phase 1 trial would need at least 15 for 5 to 10 patients to begin with, and a phase 2 would need 40, 50 patients. Do we have those patients, well documented patients with so and so gene being mutated? We don't have the data yet. So we need to start from there. So therefore, it is important to start now, if not later. So I think with that, I will stop and will be open to take questions. This is a small team. Terry Prasad, which helped us to set up all this process. Thankful to all the clinical collaborators and the collaborators whom helped us with the gene editing program. And now I'm based in STEM and open to collaborate with many of you here.
Thank you, Dr. Indumathi, for that extremely enlightening talk and very outstanding talk. I mean, in fact, I thought that was perhaps at some point in time, you could give a series of lectures to our ophthalmologists and basic science of each of the areas you've worked on. I throw open the lecture for discussion now. I'm sure there are a lot of enthusiastic ophthalmologists from various branches who would like to ask questions. And our basic science as well.
It was a great quote. So I have three questions. So when we use FN casting for the basic protein, I know it works differently compared to SP casting. It has flexible band apart from NGG. Also, the geometry of DNA binding is different compared to SP casting, and it has a broader editing windows. I'm really surprised with data. there is on target of 90%, but there is no bystander rating within the protospace. Since given that it has high fidelity and it's hyperactive base editor, but and very sufficiently there is no bystander rates.
Yeah. So the data that I've shown is the clonal data. It's a clone. In a pool analysis, if you look at the whole genome profile, about 70% of the edit were on target. Maybe 2 or 3% had off target. And bystander, that too particularly, this analysis is for the target site analysis I'm talking about. Target sites, if you see that one particular target for the RP65, apart from the target A, within the edit window, there were 3 other A's which are very likely to be target for bystander edit. We were expecting a lot of problems there at that locus, but we were totally surprised when we saw that results that more than 60% is edit, and edit out of the 60%, nearly more than 90% were on target. So the off targets were very minimal. It was there. It is very minimal. Then when we picked the clones, we picked the two clones which are positive. On those 2 clones, absolutely no bystander. Only the target site. So we ensured that the target loci has been well characterized, and then we took these 2 clones for the whole genome analysis for any off target also. So unlike SPV, which you have anywhere between 100 to 1,000 different off-target regions elsewhere in the genome, we found four sites elsewhere by genome sequencing, and 4 of these sites were pretty much in gene islands. So basically, in some intronic regions, we found.
What are the data we showed is like a clone that don't have any bystander.
Yes, exactly. So that is the clone. So after characterizing all of this, we took that clone forward for differentiation. Yeah, so that's the advantage in an ex vivo editing system. At least you can make sure the edit is correct, and then you take forward for the product development down the line. But in vivo, if you're talking about, you really don't know. So this is true for this particular target which I talked about. So it may not be the case, even with FN-Cas, it may not be the case in a different target. So
second question is regarding the RD3 corrections now. So you have developed heterozygous clone. I know it's autosomal recessive. Correcting one allele should be fine to the So you are not getting homozygous, or so why it's
no, it should be homozygous also. So we didn't screen enough, I would say. Probably the edit efficiency was low in that batch. So we happen to have a heterozygous edited clone being picked up and characterized. But if we have screened sufficiently, we would want a homozygous edit, definitely. So we would have come across. But the line that we have picked up and characterized, so that carried in heterozygous. But that also helps us to test the proof of concept, in the sense like, will in a heterozygous condition, if heterozygous single allele edit will also can revert the function or not. We could able to show that even if only one allele is edited, the splicing is restored. So it kind of helped our proof of concept too. Yeah.
So during your evaluation, you got a chance to analyze the RD3 protein because you showed only the mRNA splicing data. And since you mentioned in the early slides that it involved trafficking, like towards the receptor. So did you check this red GC trafficking?
So we have checked the protein levels, as you said, yeah, it's at mRNA. So we checked the protein levels both for RP65 edit as well for RD3. By Western, we have shown that it is being expressed. But we haven't done any extensive characterization on the photoreceptors. Like once the photoreceptors have been made, then is that RD3 protein going to the right compartment? So that would need a good antibody that will help us to validate the IF by IF also. That we had problem with the RD3 antibody, so we couldn't do that. But the functional red GC localization could be done, which we haven't done it.
Waiting for that to go.
Yes. Yeah.
Sorry, I have one more last question. So what modality used to make the IEDs? Like you mentioned lenti and
so we have tried, like we have started long back. So we started from lenti retro system to begin with. Then we moved on to episomal for some of the GMP lines manufacturing. And the latest one, which we have really that has gone to CDSCO approval for the RP product, that was a Sendai virus derived line. Yeah, CTS line, CTS and cytotune get reprogrammed line. So we have 3 lines which are well characterized from that GMP batch. So that's available for clinical use. Yes.
Any other question?
Ma'am, Anupria from Oftal, dealing with retina. Once these things are made, do we inject into animals or we directly go to patients?
No. We need to do sufficient preclinical. So these organoid models and all of that will help you to validate to some extent, be it gene therapy or any of the gene editing therapies. We can validate in the cell line models and the organoid models in vitro. Once we are sure about the biology there, then you move on to animal models. You test the proof of concept in an animal model also by injecting it subretinally. At least for the safety and also for the dosing also, how much dose, how to inject, even the process of injection, subretinal injection also need to be validated in a real situation. So there we do preclinical studies.
Yeah. We use because the mouse thing is different.
Yes. Yeah. So yeah, for many of the retinal dystrophy mouse models, genetic models of mouse models are available. But then my size are very, very small. That's a problem. Many of our clinicians would help us really find it difficult to handle because you are expert in handling the big eyes in humans. So same thing when you come to the animals, it's a very small eye, and that going into the subretinal space is a challenge. So we had a compromise there we moved from mice to rat models, just a little bigger eye. So there is a RCS rat models that are available. So if you see many of the clinical trials that have gone, so most preclinical studies were done in RCS rats. So that's available. But for the dosing and for the actual safety studies, the regulators asked for one large animal model. So one small model and one large animal model. Large animal model can be anything between pig or dog or any primate. So that's mainly for the dosing. So you're injecting at the say if you're in mice and rats, you can only go up to 2 to 5 microliter. You can't. So that's the maximal volume that you can inject in the subretinal space. But for a large and in humans, we are going anywhere between 50 to 100 and 150 microliter. So for that dose to be tested, you need a little larger number, at least for the safety of that level of dose. So that may be a small batch of 5 to 10 animals, large animals have to be done. So that data regulators would want it. We have to show that.
I'm in the cornea department. So my doubt is like in corneal endothelial cells, you're looking at that, how to culture them.
Yes.
So what do you need for that? Like, are you doing it in the patients or still in the lab or like, how are you going to proceed for that?
Yeah, so if we are looking for any endothelial disorders, so one thing is if you want a disease model, then if you know the patients who have mutations already documented, then we can generate the model and study that. That's one thing.
Patients.
Yeah. For therapy, it can work for cornea, it can work as an allogenic because cornea is avascular, right? So we have already well characterized iPSC lines available both here, CSCR, and we also have them. So we can differentiate and make endothelial cells. We are also thinking about making it as a complex graft using a scaffold. Like you in an endothelial keratoplasty, you roll this endothelium and then load it into your delivery vehicle and insert it, right? Can you make similar graft like that with IPC derived endothelium? Have a scaffold on which you make this monolayer of endothelial cells. So this is IPC derived endothelium for allograft setting. Can we make this complex graft and use it as a therapeutic product? So that's something that we are thinking of developing.
What about injecting? Some people have injected the cells. That doesn't.
Yeah, injection. Yeah, the current ongoing trial by the Japanese group, they're actually injecting a cell suspension, right? So, and they are asking the patients to lie down in a supine position so that cells by gravity it goes down and then settles on the inner surface. We don't know, but then with the aqueous humor being secreting, will that go into the trabecular meshwork and the excess cells, will that create a clogging? We don't know that. Right? Let's wait for the result, our trial outcomes. We'll know what is going to work or not work. But our approach, we are thinking of establishing a graft which is on a scaffold so that the approach of delivery will be the same as what you will normally do for the DSAC surgery.
Hello, ma'am. I have two questions here. Ma'am, this is regarding the RD3 editing. So the clone that you edited is the homozygous clone. So
The RD3 is an heterozygous clone.
Heterozygous. Then it with heterozygous clone, it showed a complete shift in the splicing. Is it?
A partial shift. You saw both normal splicing as well abnormal because you have both alleles there, normal and abnormal allele. So you saw both kinds in the RNA.
Yes. So what is the reason for that very faint band? Because it's a heterozygous clone, we would expect to see a proportional band.
Yeah, the faint is with respect to the intensity, but the size, you have to think about the size. The smaller product is a smaller size, right? So the number of molecules in that will be equally same as that of the other one.
That is something that we also see after heterozygous correction. We do expect a partial split of the intensity, but it's always fainter. So I just
it will be fainter for the same number. Say if it is a 100 molecule of 1 kb fragment versus 100 molecule of 100 base pair molecule. So the intensity will be different, but then the number of molecule, when you quantify with respect to the molecule for the size of the DNA, it will be the same. Right. So if it is the one of the product is a smaller product, you will expect it to be fainter only, right?
And
theoretically,
yes,
right.
Yeah. And the other one is what is the platform that you use for base editing, IPs?
In the sense the delivery for the so we use regular lipofectamine basis. There is a stem reagent from the invitrogen, so that works very well. And you can also do nucleofection if you're thinking about RNA or an RNP delivery, and nucleofection works better. The Lonza or the invitrogen that works well. Only thing is there you will need more amount you required for the nucleofection is more. Lipofection you need less amount of
Very nice talk, Dr. Indu. Just I have one or two questions. One is have you transplanted these cells, the cells that you showed, whether RP or other cells that you have made? Have you done any transplantation studies so far?
Yeah, not for the edited cells, but we have done proof of concept studies for a normal iPSC derived RP cells, which with that data we went to the regulators for getting the CT10 approval. So there we have done about 20-25 animals. We have done the subretinal injections, and we have followed it up to 3 to 6 months, and shown that the cells do survive in the subretinal space. So that data is available, and we know how much to inject, how to inject in the subretinal space. In the RCS rat model, we have done that. And in nude mice also, we have done just to make sure our prep is good enough, that you have more than 95% RP cells, and whatever residual 5% is there, it's not the iPSCs, which can give rise to teratoma. That regulators want to see it in a nude mice, where you inject 10 times or 20 times the normal dose. Like say if it is 2 lakhs, then you go to 2 million cells and inject in your mice and show that it doesn't form teratoma. So that data also we had, so we have done that preclinical studies in a nude mice model too. Yeah.
Okay. The other question is how long these cells survive once you transplant? Where in case of what is the data available in case of humans?
Yeah, data available in case of humans, I wouldn't know because the clinical trial reports should tell. But in animals, whatever we have seen in the 6 months timeline, people are not just us. Many reports, preclinical reports are available. The cells are there still. And if you think about human development also, both retina, most of the CNS neurons and terminally differentiated cells are made for life, right? So they are made and there's no regeneration that's happening there. So these cells survive for the rest of the life. In vitro, if you keep these RP cells in culture for 1 year, 2 years, and just keep maintaining them by changing media every day, they do survive. So they don't die. So the only catch here is in the patient, in a diseased environment, if the milieu is not conducive, there's a lot of inflammation that's already going on, and you are putting these cells in. Whether it will survive that challenge in an in vivo milieu is something that is unknown. We will get to know only with more of trials coming up. As of now, in theory, these cells should survive provided the milieu is good enough that the patients can retain them for longer time. So that's the idea. So therefore, these are considered as a one-time therapy, but don't know. So we can't answer it right
But
now.
in the animal model, if you have used any pig or any big higher animal models, how long they have seen the maximum survival, like 1 year, 2 years? Any data is available from the literature
For
in the preclinical
gene
studies?
therapy, lot of because which has started long back, people have shown that even after 10 years, the genes are there. But in cell therapy, I think people have followed up for up to 1 to 2 years. 2 years for post transplantation, people have documented them to be there, but we don't know. We don't know, not many. So the field is at very nascent state. Very few clinical trial outcomes have come out from UK, from Japan, and US. US trial started way back with ES derived cells. So there we have maybe 78 years outcome reports people have shown it. But IPCs it's very early. So there are only a handful of not even handful. I think 1 or 2 reports available at this point, and many trials going on. But we have to wait for their outcome reports to see.
Thank you very much. I think we have, okay. So one last question before.
Yeah, thanks, Dr. Indumathi. Nice talk. So, you know that these are rare disorders. So, that like you have SEP 290. So, it's a ciliopathy. So, by the time it's like a diagnostic odyssey, it will take 4 or 5 years before we reach the correct diagnosis. So, at that point, when the damage may have already occurred.
Yes.
So, will that be useful for, like, if we have to collect the epithelial cells or do something for those? Is that going to be feasible for them?
Yeah. So, if you're talking about gene therapy, cells have to be alive. Gene therapy or in vivo gene editing, whichever it is. So, if the cells are gone, then they are not the candidates for the trial. But for cell therapy, again, I wouldn't give any comment for any photoreceptor-based disease, like say ciliopathy genes are all mostly photoreceptor diseases. But if there are a condition like BEST, and AMD, and LCA2, which is an RP65. These are RP-based diseases. The cells that are affected are RP cells. They are the ones that are going to die first, and eventually photoreceptor will die. Right. So in those cases, if we can catch the patients where the RP degeneration is just starting and photoreceptors are still fine, we have a window to intervene where we can really deliver these cells. And if functional RPs are there, they should support the photoreceptors to survive. So that is the expectation of many of the RP-based cell therapies. So that's where it's important to identify who the patients are, what mutations they carry, whether it affects RP cells or photoreceptor cells, and stage of their disease progression. All of this is very important. We need to identify these and recruit the right kind of patients in for any clinical trial. So that's my important message that I really want to convey here. So because patient selection is very critical in many clinical trials.
I think in view of time, being short, I think we'll have to close the session. But as Dr. Indumathi is actually going to be with us till late this evening, and I'm sure some of you as ophthalmologists actually meeting over in the afternoon. But you're most welcome to interact with her. Dr. Indumathi, we look forward to further collaborations. I anticipate a golden triangle between Shell Hospital, Ophthalmology, CSCR, CMC, and INSTEM Bangalore as far as ophthalmological research is concerned. And I'm certain many of these ideas of yours will be translated into clinical reality. So, with these few words, I would like to encourage all of you to stand and give her a standing ovation.
Thank you.
Thank you so much, Dr. Indumathi.
I'm looking forward for a very long-term and productive collaborations too. Thank you so much.
Thank you.